Today, we are on site at the Moscone Center in San Francisco, California at the 2020 Genitourinary Cancers Symposium. We have recapped some of the top news presented each day during the meeting, and soon we'll speak with Dr. Daniel Petrolak on the key studies in bladder cancer and prostate cancer, as well as Dr. Brian Rini on the latest in kidney cancer. Welcome to OncLive News Network. I'm Gina Columbus. In advanced clear cell renal cell carcinoma, the oral HIF-2 alpha inhibitor MK-6482 demonstrated promising clinical activity in heavily pretreated patients in a phase 1/2 trial. Results showed that the objective response rate with MK-6482 was 24%, with 69% of patients experiencing tumor shrinkage. Moreover, the median progression-free survival was 11.0 months. The agent was also found to be well tolerated. Based on these encouraging data, there are plans to launch a phase 3 trial with MK-6482. The combination of the RTK inhibitor sitravatinib and nivolumab showed a benefit in patients with advanced clear cell renal cell carcinoma following progression on prior VEGF-targeted therapy. Specifically, results showed that there were higher objective responses and longer disease control versus what is historically expected with single-agent nivolumab in this patient population. The first results of the phase 2 Nevus trial showed that nivolumab in combination with stereotactic body radiotherapy led to a high disease control rate in pretreated patients with metastatic renal cell carcinoma. The regimen also led to no added toxicity, and an analysis is ongoing to explore the correlation between efficacy of the combination and PD-L1 expression. Real-world results of the MEET-Euro-7 study showed that following progression on immunotherapy, most patients were treated with VEGF-TKI therapy and mTOR inhibitors, which were found to be both active and safe. Specifically, the multikinase TKI cabozantinib was found to be associated with a longer modified progression-free survival. In the retrospective study, investigators evaluated outcome data, including PFS and toxicities, from 162 eligible patients who were treated across 16 Italian referral centers. 42-month follow-up of the phase 3 CheckMate 214 trial showed that the combination of nivolumab and ipilimumab showed superior objective response rates and overall survival compared with sunitinib in both intent-to-treat and intermediate and poor-risk patients with advanced renal cell carcinoma. Additionally, more patients who were treated with nivolumab and ipilimumab experienced a complete response versus sunitinib. Based on earlier data from the CheckMate 214 trial, the FDA approved this combination in April 2018 for intermediate and poor-risk patients with advanced renal cell carcinoma. Hi, we are here with Dr. Daniel Petrolak, who is Professor of Medicine and Urology and co-leader of the Cancer Signaling Networks at Yale Cancer Center. He's going to discuss some of the prostate cancer and bladder cancer data being presented at this year's symposium. Hi, Dr. Petrolak. Thank you so much for being here.
Thanks for inviting me, Gina. How are you?
Good, thank you. How are you?
Good.
Good. So let's start off with bladder cancer.
Right.
What would you say are really some of the exciting data that we're learning about this year?
So I think one of the more exciting presentations is an extension of something that was presented at ESMO with enfortumab vedotin. As we know, enfortumab is a monoclonal antibody that is linked to a chemotherapeutic agent, and we've seen a 40% response rate in patients who are refractory to 2 different treatments, checkpoint therapy as well as chemotherapy. So what this trial did was it combined enfortumab along with pembrolizumab in patients who were not eligible to receive cisplatin-based chemotherapy. And the objective the anti-tumor shrinkage rate was 84%, which is pretty impressive. Enfortumab by itself has about a 70% shrinkage rate overall, not partial and complete, but overall shrinkage rate. So this is really one of the more active combinations that we've seen. We're also seeing that the median time to progression is about 12 months, which is pretty impressive as well. So I'm really excited about this data. I'm proud to be a part of the trial, and we hope for more information, perhaps a randomized study coming in the future.
Very exciting, thank you. So now I'm going to jump over to prostate cancer. You're the lead author of a poster looking at the association of humoral antigen spread with cytotoxic T lymphocyte activity after sipuleucel T in the phase 2 STAMP and STRIDE trials. So can you give a little overview and discuss those findings?
Sure. So STAMP and STRIDE were trials that were looking at combinations of sipuleucel T with different hormonal agents, enzalutamide or abiraterone or prednisone. And what we did in this study was we looked at the different patterns of immunity that developed with sipuleucel T treatment. Sipuleucel T is a dendritic cell product, and that is raised against something called prostatic acid phosphatase. And there are 3 infusions that go back into the patients. We thought this was all predominantly a dendritic cell process. What we're seeing is, as time goes on, there's something called antigen spread. So the cancer cells are basically lysed. They release antigens aside from PAP. Those include PSA, PSMA, prostate stem cell antigen. So if you look at those, there's a whole variety of different antigens that come out. It turns out that later on, the cancer cells or the patients develop B-cell immunity. They develop monoclonal antibodies to those particular targets. So there may be two different ways that Sipuleucel T induces cellular death and cellular killing. So this is an important product. I think it's a little bit underused in the treatment of castrate-resistant prostate cancer, mainly because in the past people thought that, well, there wasn't a progression-free survival difference. That really didn't make sense. But now, as we're seeing with immune therapy, you don't need a PFS difference to get a survival benefit. And the IMPACT trial, which was actually now almost 10 years old, demonstrated a survival benefit for CYP-T in patients who are asymptomatic or minimally symptomatic.
Very interesting, thank you. Now, I wanted to talk about the COSMIC O21 trial that's gaining a lot of interest at this year's meeting too. So can you give some background to the COSMIC clinical trial program and then what we're seeing here?
So cabozantinib, the original use of cabozantinib was intended for prostate cancer. And cabo had some really very, very interesting patterns of response. It was showing bone scan responses and improvement in bone pain, and this is what got a lot of us excited. Unfortunately, the COSMIC-1 and the COSMIC-2 trials failed to meet their primary endpoint, predominantly because this was in a very, very refractory group of patients. Cabozantinib is a kinase, and of course kinases have, or what we call dirty drugs, they have multiple actions, mechanisms of action. And one of the actions of cabozantinib is a decrease in myeloid suppressor cells. So it has an immune modulatory effect. By itself, cabo has about a 4% response rate in prostate cancer. Not particularly impressive. Same thing with the atezolizumab. Atezolizumab, it's less than 10%. And we had actually presented some data a couple of years ago looking at atezolizumab as a single agent in castrate-resistant disease. So an interesting survival pattern. It looked like in a refractory group of patients they were living longer, but non-randomized, so you really can't claim anything from that. When you combine the two drugs together, you get about a 30% response rate, which I think is incredibly exciting. The question is why? If you look at the spider plots, actually what you see in a lot of these patients is an increase in tumor volume and then a decrease, which tells me this is probably an immune mechanism. The cancer cells or the immune cells are migrating towards the measurable disease, and then they're causing their antitumor activity and then causing the cancer cells to die. So I think that there's a lot of interesting questions mechanistically with this. It's potentially affording us another option, but think of it this way, two drugs which really have minimal activity separately now are combined together and are showing activity, true synergy.
What do you think the future holds for this regimen? How do you see it best fitting into the landscape?
Well, I see this either in place of chemotherapy or after chemotherapy. These are patients with poor risk characteristics. We have a lot of, as we're treating more patients down the continuum, we're seeing the disease get more aggressive, more liver metastases, more spread to other areas. The most impressive response in this study was a patient who had almost a complete response in liver, which is outside of platinum is not really seen. So I think that there's great potential for this, but either in visceral disease or a combination with chemotherapy or in other areas, but we're excited about it.
Very good, thank you so much. Now, PARP inhibitors have also been making some headway in select patients with prostate cancer. So this year we saw data from the combination of olaparib and cediranib in patients with metastatic castration-resistant disease. So what's important to know about these data?
So I think it's important to know that with BRCA and with DNA repair mutations, PARP inhibitors are useful for those types of patients. That's only about 20% of all patients with castrate-resistant disease. So we want to try to expand that group of patients to include others. This work was done by Joe Kim at Yale, who is one of our attendings who works in a group, and Joe's really a fine young investigator, and he really should be congratulated on this trial. What Joe did was he took patients with castrate-resistant prostate cancer. They were not selected. We're looking at those molecular markers now, so we don't have the marker data. Randomized them to receive either olaparib or cediranib. Cediranib is an anti-angiogenesis agent. And the theory behind this is that by causing hypoxia, you induce BRCA. And you induce BRCA-like changes in the tumor cells. So this makes them susceptible to PARP. And this is a way for us to expand this out. So what Joe found was when you compare olaparib cediranib to olaparib alone, there's a 7-month difference in radiographic progression-free survival. That's statistically significant. The other interesting thing from this trial is one of our patients who was treated had initially responded to olaparib. He was a BRCA-positive patient. We then put him on cediranib when he progressed, along with the olaparib, and he responded again. So it's further support that this hypothesis may be valid. It's not only being done with olaparib and stereotactic, it's also being done with abiraterone. Abiraterone will decrease the DNA repair enzymes. Same thing with immune checkpoint agents. So I think that we have really a lot of areas to explore with PARP inhibitors in terms of developing new treatments and expanding its use for patients who may not be BRCA positive.
Very wonderful insight. Thank you so much. Is there other research that you're authored on this year that you would like to highlight?
So we presented our schema for the KEYNOTE-921 trial, which is a randomized trial comparing docetaxel plus pembrolizumab to docetaxel alone. There have been preliminary reports of this combination showing an increase in radiographic progression-free survival over historical controls, and we're excited about this study. It's more than 1,000 patients internationally. We're accruing nicely on it, and so hopefully we'll have an answer in a year or two as to whether this actually improves survival or not.
That's great. Thank you so much, Dr. Petrog.
Thank
we really
you.
appreciate you being
Always
here.
Always a pleasure. Thanks.
Hello, and we are here with Dr. Brian Renal, who is the inaugural Chief of Clinical Trials at Vanderbilt Ingram Cancer Center. He's going to chat with us today about all of the exciting data in kidney cancer being presented at this year's symposium. Hey, Dr. Renal, how you doing?
Good. How are you? Thanks,
Good.
Gina.
All right, so there's a lot going on this year. One of the things that we heard about were the longer follow-up from the Checkmate 214 trial looking at Nivolumab and Nivolumab in intermediate and poor risk patients with advanced RCC.
Right.
So what are a couple of things that we learned with these longer follow-up?
So one of the nice things about this data set is that it has the longest follow-up of the phase 3. So this was called the 42-month follow-up, meaning there was a minimum of 42 months of follow-up. So we're getting out to that 3-4 year range. This is obviously an all-immune combination, and the strength of immunotherapy, especially dual immunotherapy, is durability of response. I think it's its calling card. And so the nice thing about this data set is we're starting to get this long-term follow-up. We're seeing patients who are in remission, I think, meaning they're either on or off treatment with long-term disease control. I believe the progression-free survival rate, out at the 42-month mark was something like 35 or 36%. Oh. So it means a third of patients that you start on therapy are still going to have disease control, 3.5, 4 years later. That's amazing for kidney cancer. So I think that's the real value in this data set. A lot of the other numbers didn't change, hazard ratios and PFS, and so that was all consistent. But again, I think as this data set further matures, we'll see more and more durability.
Good.
Yeah.
And now we also have the final analysis from Checkmate 025 looking at single agent Nivolumab.
Right.
So anything different that we learned with this data set?
Not much, and as you know, at least in the US, there's not much single agent Nivolumab used because it's used up front, but it's nice again to have that long-term follow-up. I believe if I read the swimmers plots right, that there were, trying to remember the number, I think it was 10 or 15% of patients who were off treatment and never received further treatment and who are still alive. So again, if you think about that, 15% of the original population. So again, speaking to immunotherapy that you can start people and there will be a subset who's just gonna have really long-term benefit. I think it's greater with dual immunotherapy or the IO-TKI combos. So it's somewhat of a historic data set, but it still, again, speaks to the value there.
Great.
Yeah.
Now, we also saw a lot of a newer agent, the HIF-2 alpha inhibitor, MK6482 in advanced clear cell RCC. So could you discuss this agent and some of the activity we saw with it?
Sure. So this agent's had about 3 or 4 different names, so it's hard to keep track. But so this was originally a Peloton compound. Peloton was acquired by Merck recently. That's how it switched names. So this is the second generation HIF inhibitor. It's an oral drug that inhibits HIF, obviously, which is a transcription factor crucial to the biology of kidney cancer. It's really one of the first examples, maybe the first example of successfully targeting a transcription factor in oncology. Nobel Prize was just awarded for this. So basically, this was a trial in refractory kidney cancer, and they've produced now a series of about 3 trials in refractory kidney cancer. And all of them have a very consistent theme. In fact, the discussant, I think, put them all on the same slide, nicely sort of summarizing the response rates in a really highly refractory 3 or 4 prior treatment group was in the 20 to 25% range, and that's been pretty consistent. The other important characteristic of this drug is that it's really well tolerated. So VEGF TKIs, which we use as monotherapy and in combo, are great drugs, but not always well tolerated, as you know, and sort of come with their own chronic side effects. And so this drug has activity, but it's really well tolerated long term from the data from my own personal experience. So there's a registration trial going on now in these refractory patients that hopefully will lead to approval. I think more exciting is that it can be a component of doublets and triplets. And as we're starting to combine more and more drugs to hopefully cure more patients, we have to be mindful of toxicity, right? It's easy for us to just start combining things. For patients, not so easy. So if this can be a VEGF, TKI-like drug with less toxicity, that would be a major advance.
Yeah, that's some great points.
Yeah.
Now, there's a couple combination studies also with Nivolumab that were presented. So could you share insight on the Nevus trial as well as the phase 2 trial with Sitravatinib and Nivolumab?
So Nevus, so there were a couple trials, Nevus being one of them, that looked at radiotherapy in combination with Nivolumab. Hans Hammers presented one of radiotherapy with Ipi-Nivo, same theme. So this is stereotactic body radiation therapy, or SBRT, which is over the last, I don't know, probably decade, maybe not even, it became useful in lung cancer, originally tested in elderly patients who couldn't get, a thoracotomy. And so it was shown to be effective, and now it's being used against metastatic sites, including kidney cancer. The theory is that by radiating the tumor, you get antigen release, and that'll increase the immune response when you give an immune agent and lead to more systemic anti-tumor immunity. I think the trials, and Tom Powell's did a nice discussion, I think it's not clear from the trials that the activity seen was any more than you would have expected from the immunotherapy alone. So I'm not convinced that it's a way to increase anti-tumor immunity. Having said that, I use SBRT in my own practice. So an example would be a patient that I'm observing who then has one area grow, still asymptomatic. I'm not ready to give them systemic therapy, so I might use SBRT in an attempt to defer therapy. That might be one example. Okay. Patient who's on systemic therapy responding but has one or two areas grow. So I think the field is figuring out what to do with SBRT. I use it more and more, although I have to tell you I always struggle when I'm giving it to a patient to ask myself, am I really helping this patient? Am I making them live better or longer by doing this? And I'm not sure there's definitive data. So interesting studies. I think more work needs to be done. There's, I think, at least one randomized study being done. But it's a new tool to play with.
Definitely.
Yeah. And then the other one you asked about was Sitravatinib, which is a multi-targeted agent with targets similar to cabozantinib, I believe, VEGF and MET and other things, in combination with Nivolumab, I believe, in a refractory setting. And that showed activity that I would expect, right? We know IO plus TKI is active, right? There's no question about it. Again, one of the fundamental questions for the field is, are the different targets and properties of TKI, do they affect immunomodulation differently? So if having MET and AXL and TAM inhibition better than just having VEGF inhibition, better than having whatever, I don't think there's great data. There's not great clinical data to show that. In fact, the discussant showed a slide really of all the major VEGF IO combos or TKI IO combos, and activity looked pretty similar to me, within the comparisons of small trials. So it's interesting. I don't know where it's going to go development-wise in kidney cancer. It's a pretty crowded field, but we'll see. But interesting, I think, supportive of what we already know.
Good problem to have. A lot of combinations showing efficacy. Yeah. And speaking of which, last year we saw the approval of Nivolumab and Axitinib in the frontline setting for advanced RCC, and this year we saw some more analyses from the Javelin Renal 101 trial. So can you discuss anything that we may have learned from these studies this year?
Yeah, so Javelin's done a nice job of looking at their data in different subsets with regard to nephrectomy or not. I believe at this meeting there's a gene expression, some of the translational, and we're really, we have these big clinical pieces, as you mentioned, that have led to FDA approval, and we're really just starting down the road of understanding the biology of response. One nice thing, so there's data from the emotion study that Dave McDermott published in Nature Medicine. Javelin has looked at that signature and other signatures, and there seems to be a pretty consistent theme that kidney cancer is, not surprisingly, driven by angiogenesis biology and inflammatory T-cell biology, and likely some others. But I feel like we've learned more about the biology of kidney cancer in the last few years than we knew because of these large-scale gene expression studies. And I give kudos to the sponsors, to the companies who've, spent time and resources doing this, even if it doesn't necessarily help their own regimen, because we're not selecting therapy based on it. It's not a clinical biomarker, but I feel like it's making us smarter about the biology of kidney cancer.
Good, great insight, thank you. Now, is there other research that you are involved in this year that you would like to highlight?
So, I'm involved in a lot of things. I guess just to maybe broaden that and say what's the field sort of waiting for, doing. So there's, 2 more phase 3s, Cabo-Nivo and Len-Pembro. We're waiting for those to come in. My expectation is that they'll perform like the other TKI IO combos. Whether or not they'll have a survival advantage, it remains to be seen. And, I don't know that they're going to necessarily be in advance. It's hard to believe they're going to be that much better than, say, Axi-Pembro, which we have. But we're waiting for that. And then we're kind of resetting and saying, okay, what's the next wave of phase 3s here? We have this HIF molecule that's exciting, going to start to be integrated, and obviously was bought by Merck, who has a lot of resources and drugs to develop it. We're waiting for these adjuvant trials to read out, which I don't know will be this year, but should be in the next year or so, which could then totally change the landscape and the kind of frontline patients we see. So we're kind of in this transition period where one wave of phase 3s are ending, and I think another one's about to begin.
Nice. And then, speaking of lenvatinib, actually, can you maybe expand on some of the ways that we're best using this agent, and if you want to expand a little bit on the pembrolizumab and lenvatinib, or even looking at it in the second-line setting with everolimus?
Sure. So lenvatinib has been studied in combination with pembrolizumab. We usually call it len-pembro. And as I mentioned, there's a big frontline phase 3 trial that should read out, and we all expect activity. It had some of the most impressive phase 2 activity. In the second line setting, it's been used with everolimus as sort of in the refractory setting. That combination of len-everolimus is also an arm of the frontline study. So and I think it was done as a post-marketing commitment after they got approval. And so it'll be interesting to see how that compares with the len-pembro. My expectation is that len-pembro will be better. I'm a fan of putting IO up front. That regimen certainly has utility. It also has some toxicity concerns. And so where that regimen's going to fit in, I'm not actually sure. I think doublets and triplets that are IO-based will dominate the front line, and then we'll start to sort out the salvage setting. And whether immune therapy is active after immune therapy is a big question. So I think it's, the role of lenvatinib and those combinations is sort of evolving. Len-everolimus in the refractory setting, len-pembro in the frontline setting, but we're waiting for some of these big data sets to sort out. And then just as one more side note, there was some data presented at ESMO about len-pembro in patients who were refractory to PD-1, PD-L1 inhibitors. And I don't remember all the details, smallish single-arm study, but the response rate was something like 60%. Wow. So again, a question for the field is IO active after IO? And this would suggest yes. Again, single-arm study, small numbers, all the caveats, but it's an area where I think you'll see a lot of drug development in the future in trials.
Thank you.
Sure.
Now, I also want to turn over to your Amigos, the Twitter group that you're very involved in. So can you tell us a little bit about what you really want to accomplish with that group, who's involved, and what do you hope the oncology community can take away from it?
Yeah, thanks for asking. So your Amigos. So this started probably 18 months ago. Tom Powles and Chris Sweeney and I decided that we wanted a presence on Twitter because there were a lot of people on Twitter and we thought it was important, but we wanted it to be an educational scientific presence, not look who I ran in at the airport, I'm going to post a picture presence, which is a lot of Twitter. And so we started the group and started to tweet and then have expanded it. Dave McDermott, who I mentioned, is also part of it. And then Silke Gillison, who's a prostate cancer expert in Switzerland, Kala Sridhar, who's at Princess Margaret in Toronto, Laurent Salbiget, who's at IGR in France, and Cristina Suarez Rodriguez, who's in Barcelona at Vall d'Hebron. So it's a very diverse geographic, gender diverse, everything diverse group. And we started to branch out. We've actually started doing some podcasts recently. So over the last week or so, we've done a bunch of podcasts. Tom and I just did one downstairs around the renal posters.
Cool.
Yeah, we're all a little bit technically challenged, so we need some help figuring things out, but we're getting there, and it's just fun. It's a fun thing to do. But we think there's a need for it. There's surprisingly little of that, in terms of sort of education. We've gotten a lot of good feedback. People like it. And so I think we'll go out, continue with the podcast, We're open to ideas. If people have ideas, I think we'll probably start doing some like Twitter online tumor boards. There's some other non-oncology fields that do that. Bring in some patient advocate groups. It's a great way to get them involved in sort of the CME discussion. So it's been a fun thing, and it's kind of taking off. So we'll see where it goes.
You've been doing a lot of tweeting as well during the meeting and polls and.
A little bit. Yeah. One of the reasons we all banded together is that individually we knew we couldn't do enough. I think to be relevant on Twitter, you have to do it 3 or 4 or 5 or 10 times a day, and it just wasn't happening. Mm-hmm. So I would say we go in waves. I did during the session this morning. We did a bunch, and then it'll be quiet, and so. None of us are really like sort of super users or whatever the right term is, influencers. And so we need to do a better job at that. And I'm hoping the podcasts and the Twitter tumor boards and stuff, are a way to expand our presence besides just, brute force tweeting, which is kind of what Twitter is, right? You have to brute force tweet in order to be relevant. So we're trying to at least do different things. We'll see how it goes.
Very multi-pronged approach.
Yeah, hopefully so.
And people can follow you at
Your Amigos. Yep, is our Twitter handle. So we're up to actually 1,300 followers as of this week. So
congratulations, that's great.
We're aiming for maybe 2,000 by ESMO or something. We're trying to set goals for ourselves. But
that's good. Yeah. It's doable, definitely. Yeah. All right, well, great. Thank you so much for coming, Dr. Rini.
Sure.
We really appreciate it.
All right, thanks, Gina.
Thanks. That's all for today. Thank you for watching OncLive News Network on location at the 2020 Genitourinary Cancers Symposium. Signing off from San Francisco, California, I'm Gina Columbus.